All right, next up in our series of this Unit 1 Chemistry of Life is we're talking about functional groups. So functional groups are small groups of atoms with specific chemical properties. So these functional groups can confer these properties to larger molecules whenever they are attached to them. So they can make something polar or change how it interacts with other things, right? So one biological molecule may contain many functional groups that determine molecular shape and reactivity. So we have several different ones that we need you to know. So highlighted in yellow are the 7 functional groups most commonly found in biological molecules. The R in these structures represents the rest of the molecule, so that's just whatever that's going to be. The functional group is what is in yellow. So you need to know these functional groups, right? Just try to memorize them. We're going to have activities where we try to help you with that, but these are really important that you know them. So the first one is going to be hydroxyl. So you can see we have in this column right here the class of compounds and an example. You'll find these in alcohols. We have an example here of ethanol. So the properties of hydroxyl are that they're polar. So hydrogen bonds with water to help dissolve molecules. This enables linkage to other molecules by condensation, and we'll talk about condensation later. The second functional group are aldehydes. So a C double bonded to an oxygen, and then a single bond to a hydrogen atom. So aldehydes, we have an acetaldehyde right here. So the carbon double bonded to an oxygen group is very reactive. This is important in building molecules and energy releasing reactions. The next one are ketones. So we have a carbon double bonded to an oxygen. So we have ketones, and an example here is an acetone. So a carbon double bonded to an oxygen group is important in carbohydrates and in energy reactions if it's in this arrangement. Next one up are carboxyl groups. So a carbon double bonded to an oxygen, single bonded to a hydroxyl. So we have carboxylic acids. This example is acetic acid. So these are acidic. These ionize in living tissues to form COO and hydrogen ions. So these will enter into condensation reactions by giving up a hydroxyl. Some carboxylic acids are important in energy releasing reactions. Next one up are amino groups. We have a nitrogen bonded to a hydrogen bonded to another hydrogen. So these are the amines, also known as amines. Example here, methylamine. So this is basic, can accept hydrogen ions in living tissue to form NH3+, and this enters into condensation reactions by giving up a hydrogen. Next one up that you should be familiar with are phosphates. So we have a phosphorus double bonded to an oxygen up here, and then a single bond to an oxygen, single bond to an oxygen, single bond to an oxygen. So these are organic phosphates. So these are acidic. They enter into condensation reactions by giving up a hydroxyl. When bonded to another phosphate, hydrolysis releases much energy. Last group are sulfhydryls. We have a sulfur and a hydrogen. They all can be collectively grouped as thiols. So by giving up a hydrogen, two sulfhydryl groups can react to form a disulfide bridge. This is really important in protein structure. It helps stabilize protein structure. So I have to get familiar with those and try to memorize those. Maybe do like a little table like this and try to memorize them. We'll get back into functional groups later on, but that's kind of our introduction to them right now. Moving on to topic 1.3, this is the introduction of biological macromolecules. So essentially all we need to know here is that monomers, remember monomers if you don't remember that stuff, get a different color, monomers, it's a single unit of a larger structure. So monomers have properties that allow them to bond and connect to other monomers in macromolecules, right? And we have the macromolecules right here. Proteins, those are formed from the combination or different combinations of 20 amino acids. Carbs, those are formed by linking sugar monomers or monosaccharides to form polysaccharides. Nucleic acids, those are formed from 4 kinds of nucleotide monomers. And then lipids, these are non-covalent forces maintain the interactions between lipid monomers, and lipids are a really broad category of different types of molecules, and we'll get into that. So how does this relate to this chemistry of life business? Getting really into the chemistry aspect of it. is basically the processes by which these are created, which are hydrolysis and dehydration synthesis. Both of these processes are used to cleave, to break bonds, and to form bonds between the monomers to create these polymers, right? These macromolecules, these big molecules we're going to talk about soon. So just in case you don't remember, dehydration synthesis and hydrolysis. So polymers, remember, many repeated units are formed and broken apart in reactions involving water. So a condensation reaction, this is another name for dehydration synthesis. So you should try to get comfortable recognizing whenever we say condensation or whenever you read condensation that it means the same thing as dehydration synthesis. So this is essentially the removal of water so that monomers can be linked together to create a polymer or a larger molecule made out of monomers. The opposite reaction is going to be hydrolysis. This is the addition of water so that you can break polymers into monomers. So poly to mono. And then dehydration synthesis is mono to poly. All right, and we can see this here with some pictures. This is dehydration synthesis or condensation reactions. So we have a monomer here, monomer A, maybe that's monomer B, monomer A, and then monomer B, A plus B. You can see that we have to move this hydroxyl group out of the way. So this needs to leave because it's in the way. And this hydrogen needs to leave because it is also in the way. And when those come together, that's going to form two H's and an O, which is a water molecule. So once those are gone, now we can create this bond between monomer A and monomer B. Right? We can continue this process again and again. So again, we can see we're adding this now dimer, it's 2 units, to a monomer over here. So this is C, right? So again, same idea. This hydroxyl is in the way, needs to go. This hydrogen is in the way, it needs to go. And when those come out, that's going to make another water molecule, right? And now you have A, B, and C linked together. So dehydration synthesis links monomers into monomers, and in the process produces water. The opposite reaction, like we said, was hydrolysis. This is breaking the polymers into individual monomers, and this consumes water. This needs water so that it can occur. So if we want to break this bond right here between A, B, and C, we need to add a hydroxyl group, and we need to add a hydrogen to B. So we add water in, water goes in, and it breaks this monomer off. Same deal, if we want to get B broken off from C, we need to add a hydroxyl to one side, and we need to add a hydrogen to the other side. And then there you go. After it's done this, then we have 3 monomers separated due to the water molecules that were added.